DNA replication occurs in several steps that involve multiple proteins, which are called replication enzymes as well as RNA. DNA replication is vital for cell growth, repair, and reproduction in organisms. There are three main steps in DNA replication. First, the initiation. Second, the elongation. Third, the termination. So let us first start with the initiation process. In order to fit within a cell's nucleus, DNA is packed into tightly coiled structures called chromatin, which loosens prior to replication or before the replication starts. This is to allow the cell replication machinery to access the DNA strands. During initiation, the DNA is made accessible to the proteins and enzymes involved in the replication process. There are specific chromosomal locations called origins of replication where replication begins. In some eukaryotes, like yeast, these locations are defined by having a specific sequence of base pairs to which the replication initiation proteins bind. In other eukaryotes, like humans, there does not appear to be a consensus sequence for their origins of replication. Instead, the replication initiation proteins might identify and bind to specific modifications to the nucleosomes in the origin region. Certain proteins recognize and bind to the origin of replication and then allow the other proteins necessary for DNA replication to bind the same region. In other words, the first proteins to bind the DNA are said to recruit the other proteins. Two copies of an enzyme called helicase are among the proteins recruited to the origin, as shown in the video. Each helicase unwinds and separates the DNA helix into single-stranded DNA. As the DNA opens up, Y-shaped structures called replication forks are formed. Because two helicases bind, two replication forks are formed at the origin of replication. These are extended in both directions as replication proceeds, creating a replication bubble. There are multiple origins of replication on the eukaryotic chromosome, which allow replication to occur simultaneously in hundreds to thousands of locations along each chromosomes. On the other hand, prokaryotic chromosomes only have one origin of replication. The single-stranded DNA binding protein, or SSB protein, binds to the newly or now single-stranded DNA, preventing the separating strands from joining again.
The enzymatic process of elongation is essentially the same in prokaryotes and eukaryotes. The differences are only in the complexity of the DNA molecules, in which eukaryotes have multiple replication forks as opposed to a single replication fork in prokaryotes. During elongation, DNA polymerase, which is an enzyme, adds DNA nucleotides to the 3' end of the newly synthesized polynucleotide strand. The template strand, on the other hand, serves as basis to what nucleotide the polymerase will add to the new strand. Only complementary nucleotide to the template strand is added to the new strand. DNA polymerase has grooves which allows it to bind to single-stranded template DNA. For instance, when DNA polymerase meets with a cytosine on the template strand, it adds guanine on the newly synthesized strand. This happens to each individual nucleotide until the DNA polymerase reaches the end of the template strand. However, DNA polymerase cannot synthesize new DNA strand. Instead, it adds nucleotide to the 3' end of an existing synthesized strand. To synthesize new polynucleotide strand, it needs to be initiated by a specialized RNA polymerase called primase. Primase starts the polynucleotide synthesis by creating short RNA polynucleotide strand complementary to the template strand. The synthesized short strand of RNA is called primer. Once the RNA primer is fully synthesized, primase leaves the template strand. Eventually, the RNA nucleotides are removed and replaced with DNA nucleotides. At the end of replication, the daughter molecules will now contain full DNA strands with no RNA portions. Due to limitation of DNA polymerase to synthesize only in the 5' to 3' direction, two newly synthesized strands grow in opposite directions because of the antiparallel structure of the template DNA. The leading strand is synthesized continuously towards the replication fork as helicase unwinds the template double-stranded DNA. On the other hand, the lagging strand is synthesized in the direction away from the replication fork and away from the DNA helicase unwinds and is synthesized piece by piece, 1000 to 2000 nucleotides in prokaryotes and 200 to 250 nucleotides in eukaryotes. That is why it constantly encounters previously synthesized new strand. These pieces are called Okazaki fragments discovered in 1968 by Ryuji Okazaki, and each fragment begins with its own RNA primer. These fragments are then connected by an enzyme called DNA ligase, which connects two strands of DNA together by forming a bond between the phosphate group of one strand and the deoxyribose group of another. With that, the elongation process of the DNA replication ends.
Let's talk about termination. Termination is the final stage in a DNA replication. In the case of prokaryotes, termination occurs when the two replication forks, as we can see here, meet in one point. This point is actually the opposite point of the replication origin. When these forks fuse, they allow the separation of the two parent DNA strands, rendering two circular DNA molecules as product. In the case of eukaryotes, termination occurs when multiple replicons, this bubble-like structure we can see here, meet and fuse to each other to create a bigger replicons. When all these replicons converge, then the two linear DNA molecules finally separate. Let us take a little closer to the junction to which the replicons or replication forks are destined to meet. This model right here represents the portion that is not fully synthesized with DNA strand. This can be applied to both prokaryotes and eukaryotes. As we can see right here, DNA polymerases, the green rings, are about to complete the addition of nucleotides in both strands from both directions. When all these empty nucleotides are paired with complementary nucleotides, the DNA strands separate from each other. In eukaryotes, DNA polymerase I cleaves off RNA primers, represented here in brown, and it fills in the gaps with corresponding nucleotides that are complementary to the template strand. This happens in leading strand and more frequently in the lagging strand because of the Okazaki fragments, which is formed between primers. The removal of primers and addition of corresponding nucleotide is the same in eukaryotes, but different enzyme is involved, that is, DNA polymerase beta. After all the primers have been replaced by the nucleotides, an enzyme called ligase glues these fragments together to form a continuous DNA strand. After this separation, the newly synthesized DNA molecules spontaneously turn into helical structure. It is important to remember that as soon as a nucleotide base pair are formed between the old strand and the new strand, the double-stranded molecule immediately configure into a helical structure. That is, if the DNA polymerase has passed these parts. The termination stage in prokaryotic DNA is a straightforward process, while in eukaryotes, a problem arises in the telomeric ends of the DNA. If we follow our understanding in the necessity of RNA primers to be attached first to a DNA template strand before a nucleotide can be synthesized, then we begin to realize the problem in the synthesis of nucleotides in the end of the lagging strand. This strand represents the template strand that is lagging. The direction of this strand is 3' to 5'. So the direction of the synthesis of the complementary strand is from 5' to 3' direction. Now, we realize that there will be no more nucleotides in a DNA template for RNA primer to attach. Hence, DNA polymerase cannot just add nucleotide here. We know that they can only add at the 3' end of the existing nucleotide, or ribonucleotide. When this happens, the unpaired nucleotide will eventually cleaved off, and if the DNA replication repeats, the process will degrade the telomeric region of the DNA, causing it to shorten over time. In embryonic cells, there is a certain enzyme called telomerase which helps the remaining unpaired nucleotide to be filled with appropriate nucleotides. This enzyme is a ribonucleoprotein, meaning it has an RNA in it. This enzyme binds to the end part of the DNA template and allows the pairing of nucleotide in this portion. That is basically what happens in termination.
Red.
Tagdas ang red.
Oh, hi! So today we're going to discuss the process of DNA replication. And in this portion, we're going to highlight the similarities and differences of the replication process in eukaryotes and prokaryotes.
So to start it off, we will discuss first the first stage of DNA replication, which is the initiation process. Joanna?
So actually, hi,
Hi.
before the cell reproduces, the genetic material should be replicated first. Hence the process DNA replication.
So, yes,
Yeah.
so as we've seen in the video, DNA replication is the process by which DNA makes a copy of itself during cell division.
Yes, and on the other hand, the first step of DNA replication is the unzipping or unwounding the double helix structure of the DNA molecule. And this is called the initiation process.
How does the initiation process actually start?
So there are these proteins and enzymes that are involved in this biological activity of the DNA, and they are called initiators.
Yep, basically, these initiators bind to the DNA molecules.
Yes, specifically to a sequence called origins of replication, where replication begins. Prokaryotes have only one origin of replication, while in eukaryotes, they have multiple origins of replication.
Oh, so these certain proteins recognize and bind to the origin of replication and then allow the other proteins that are needed for the replication to bind in the same region. The first proteins to bind the DNA are said to recruit the other proteins, like two copies of an enzyme called helicase are among the proteins recruited to copy the RNA.
Yes, you're right there, Renz. And in addition, each DNA helicase unwinds or unzips and separates the DNA helix into single-stranded DNA. SSBs, or the single-stranded binding proteins, stabilize the unwound DNA.
Yes, Regina. The unzipping was clearly seen in the um, video we watched, and the DNA molecule opens up, and the Y-shaped structures called the replication forks are formed. Because two helicases bind, two forks are formed at the origin of replication. These are extended in both directions as the replication proceeds, creating a replication bubble, or also known as replicons.
Exactly, Ryan. Ang cute ng replication bubble. So when we talk about eukaryotic chromosomes, hundreds to thousands of locations along each chromosome allow replication to occur simultaneously or at the same time.
Unwinding the DNA causes the DNA to twist and coil to itself. So this phenomenon is called supercoiling. And this strain is relieved by the topoisomerase enzyme.
Yes, because if not, then the strain would quickly break the covalent bonds along the DNA.
Yes, making new DNA strands starts with making an RNA primer with RNA nucleotides and primase enzymes. And the next part of the DNA replication is the elongation.
Elongation.
So, have you seen my video about the elongation process?
Yes.
Yes. It was very elaborate.
Thank you.
Yeah.
So essentially, there are no virtual differences in the elongation processes of replication between prokaryotes and eukaryotes. Only in the number of replication forks and the number of polymerases do they differ.
Yes, as explained previously, the elongation process is about the lengthening or the polymerization of new synthesized DNA polynucleotide chain.
Oh, and additionally, the RNA primer created by the primase is also extended, although it would not be used in the replication process.
This is very interesting. I would like to know about the history of it and the discovery of the Okazaki fragments. What is
So the
Okazaki
Okazaki,
fragment?
oh, sorry, so the Okazaki fragments were discovered by Reiji Okazaki in 1986. It is by the DNA they collected at the bottom of their container, which is undergoing replication. So they observed that there were differences in weight between the newly synthesized strands, which led them to conclude that the lagging strand creates DNA in fragments.
Are there any more information that we should know about the elongation process, Jasper?
Well, for starters, you can read more about the different sets of DNAs of prokaryotes and eukaryotes. Then you can research about mutations caused by mistakes in the replication process and see if any of them are related to the elongation.
Is it true that mistakes can still occur in the elongation process?
Well, Regina, yes, it's true. That is due to the high-speed process of DNA replication. But the DNA has its own repair system which checks the DNA nucleotide base pairing and correct any mistakes pre and post replication process.
Wow, that is amazing. Our body is awesome.
Yeah, so DNA polymerase halts when it reaches a section of the DNA template that's already been replicated and starts the process of termination.
Yeah.
So let's begin the termination. So the termination process of DNA replication is the end of the phase. Here we now have the newly synthesized daughter DNA strands containing half of the original template and half the newly synthesized DNA strand.
Mm. So, are there or are the differences in the termination process of eukaryotes and prokaryotes significant in terms of structure,
Well,
Ryan?
as what both of you said in your explanation in initiation and elongation processes, the complexity of DNA strands between prokaryotes and eukaryotes is the reason for their differences. The number of replication fork and shape of the DNA strands plays a lot in terms of the replication speed. Additionally, eukaryotes have RNA primers in their DNA replication process.
So Ryan, you have mentioned earlier in the video that eukaryotes have a unique problem replicating their ends, which are resolved by the enzyme called telomerase. Will you expound more about how these telomerase and aging relates to each other?
Okay, so after discovery in the 1980s, telomerase gained a reputation as a fountain of youth. Cells that undergo cell division continue to have their telomeres shortened because most somatic cells do not make telomerase and eventually prompt them to stop dividing and die. This essentially means that the telomere shortening is associated with aging. Telomerase prevents this decline in some kinds of cells, including stem cells, by lengthening telomeres, and hope was that activating the enzyme could slow cellular aging. So limited
Hello. Yeah.
cells have this
Mm.
telomerase enzyme.
Kung maka-research pa ni Light. Okay, so back to the topic.
Yes.
Let's discuss about the two general features of the chromosomal replication that we haven't mentioned in the video earlier.
Oh, so those two general features, right? So yes, if it's conservative, semi-conservative, or dispersive mode of replication, and whether it follows a unidirectional or bidirectional mechanism of DNA strand growth. So Ryan, can you please expound
Yes.
more?
Yes. So in the semi-conservative model, the two parental strands separate and make a copy of itself. After one round of replication, the two daughter molecules each comprise one old and one new strand. After two rounds, two of the DNA molecules consist of only one new material, while the other two contain one old and one new strand, hence semi-conservative.
Yes. And in addition, we have also the conservative model wherein the parental molecule directs synthesis of an entirely new double-stranded molecule, such that after one round of replication, one molecule is conserved as two old strands. This is repeated in the second round, while yung dispersive model material in the two parental strands is distributed more or less randomly between two daughter molecules in a model shown here. So, mag-insert tayo yung so all material is distributed symmetrically between the two daughter molecules. So, other distributions are also possible.
Yes, the semi-conservative model is intuitively appealing model because separation of two strands provides two templates and each of which carries all the information of the original molecule. So it also turns out to be the correct one as what Massel, Son, and Stahl have found in 1958.
So that must be a great achievement for them. So, okay, let's continue to discuss the two mechanisms of DNA strand growth. So, as far as I've learned, bidirectional replication have one origin and two replication forks. So, it involves replicating DNA in two directions. At the same time, resulting in a leading strand where replication occurs more rapidly and a lagging strand where replication occurs slower. So while on the other hand, unidirectional may involve two origins with two growing ends or one origin with one replication fork.
Yeah, very well said, Ren. So, I have a question here. Why bother studying DNA replication?
Interesting. First, fidelity. It's important for replication to have high fidelity. Mistakes in replicating DNA cause mutations that can be transferred from one generation to the next. It needs to be accurate. Second, regulation. Eukaryotes have multiple origins of replication and multiple chromosomes. They need to replicate all the DNA, but only once need tight control. Tied to the cell cycle control.
Ooh, and one more thing, its machinery is impressive, isn't it?
Yes.
Yes, absolutely. So, I guess we have tackled DNA replication at our best. So, thank you so much for your time,
Thank you for your time as well.
guys.
Bye.
Annyeong!
Bye.